So the topic of fertility draws particular attention to the female, where we've become familiarized with terms such as IVF, the journey of surrogacy, and the ways in which a woman can conceive and carry a child through alternative methods. But what isn't widely known is that 20 to 30% of males are responsible for all fertility factors. And it is of these cases that 50% of the causes are undetermined in regards to male infertility. So the issue becomes, what are the possible identifiable factors in regards to infertility in males overall? So we focus in on a model organism, for this allows us to understand the issue. We specifically start small, where we look at C. elegans because they're advantageous for a number of reasons, such as their size, their transparency, and their generation time. Because of these advantages, we can then look closer at genes that are responsible for the expression of fertility factors and what it is in particular that allows these genes to be expressed. So as we can see, we look at a histone, specifically HTOS1, which is found in C. elegans. And what histones do is they are proteins that act as spools that wind DNA, are important in the role of gene expression. And so HTOS1 is localized in the germline, and it gets incorporated during late spermatogenesis, which is basically the creation of sperm cells in males. So it has been determined that HTOS1 is associated in the function of fertility and spermatogenesis, and it helps to regulate the expression of sperm-specific genes. What's also important to note is that mutants lacking HTOS1 have a decrease in progeny count with a 35% reduction, or they have complete sterility. So from there, we want to look more specifically at what it is about the gene that allows this incorporation for spermatogenesis. So we look at HTOS1 at different domains of the gene, specifically the N-terminus, the body, and the C-terminus. And from there, we can specifically see which part of these regions will allow for the function of fertility to be specific in the model that we are researching in the lab. And from there, we can broaden this information further and look at it at larger scale organisms such as humans. And we can see in ways that which the 50% of undetermined causes could potentially lead to, where activation of genes in humans regarding sperm production and how it is we can make those unidentified cases identifiable. Thank you.